If we were to classify the groups of living things, one would notice a group of animals which are bunched together in a group known as the deuterostomes. This would not include sponges, jellyfish, most worms, such as flatworms, annelid worms, the segmented worms, roundworms, would not include arthropods or mollusks, and it would include all of the vertebrates. Interestingly, however, you might notice that it also includes this invertebrate called a landslip that's known as a chordate, and this invertebrate known as an acorn worm. It would also include starfish, sea urchins, sand dollars, etc. Why? Well, there are traits which tend to unite the deuterostomes, and I'll clarify that in a second. One of them is that one of the ancestral features of the group, going back to the early fossil members, seems to be slits in the throat. Acorn worms have slits in the throat, so did some of the fossil members, and obviously this then becomes very important for gills, as I will mention later. Where there is a heart and a nerve cord, the heart tends to be on the ventral side. Not in all of them, like a couple of these would be exceptions, but the heart tends to be on the ventral side, and the nerve cord seems to be tends to be on the dorsal side, while in all of the other groups of invertebrates, that is the opposite condition. Some of the main features which are used to group these deuterostomes end up being embryological features. So when embryos start to develop, so acorn worms also have a homologue of the thyroid gland. A lot of the features then tend to be embryological features. So we could ask the question, when an embryo is a ball of cells, it will develop a pore. And then this first pore, what does this then become in the future embryo? In some, this becomes the mouth. And the Latin term for mouth first, is protostome. And so the first embryonic opening, if that ends up being the opening which in the adult is the mouth, then that is the protostome development, which would be true of, say, arthropods and mollusks and annelids, but not of the vertebrates and the other deuterostomes. In deuterostomes, and I'm going to have to clarify this, the first pore then potentially becomes the anus. So a protostome embryo, the first pore becomes the mouth. A deuterostome, it potentially becomes the anus, although in the vertebrates, then this pore ends up being revised, and then a second opening becomes the anus. But the point is, for the vertebrates, that the first opening is not the mouth. There are also other differences we could look at in how the cells divide in the embryo, in whether the cells pick a cell fate early, whether they become one later. And then, so there's a number of embryological features which group the deuterostomes together. So deuterostomes seem to be a group more closely related to each other than the protostomes. And one could include a lot of genetic analysis in this as well. But then obviously when it comes to classifying things, picking one trait is sometimes a problem. So if you want to say having 4 legs is a defining feature of the tetrapods, which literally means 4 legs, great. However, snakes have lost theirs. Whales have lost the hind legs. Caecilians are amphibians which have lost theirs. Some lizards which have lost theirs. So is it a defining feature? Yes, it was, and it was in the ancestors, but it was then modified. Is having hair a defining feature of mammals? Yes, it is. But then whales have lost virtually all of those. You could say, well, in embryonic development, maybe they have a few. The point is, sometimes it's just a little more complex because things change over time. And this feature, which you feel is important in defining the group, that may be, variable to some degree. And so, therefore, having the first hole develop into the anus is the trait in the echinoderms and the hemichordates. But also, if you'll notice, it's typical in some of the invertebrate groups, which we call protostomes, and even a couple of the arthropods, right? If you ask, well, invertebrates, a second anal opening develops after that first one. Well, that's true of vertebrates, but also one of the groups which we call protostomes. If you refer to the body cavity, it's a coelom, but coeloms can form in different ways. And in general, the protostomes have a different type of coelom than the deuterostomes. It is determined by the mesoderm and how it forms. But if one were to look at the type of coelom that vertebrates have, in blue, that's also in a couple of the protostomes. Here's the typical coelom that the protostomes have, but then a couple have lost their coelom. So the point is that when we classify things, sometimes, especially if we're going way back in time, and especially dealing with invertebrates, where the last common ancestor was rather simple, sometimes we're summing together a lot of traits, a lot of genetic evidence and a lot of separate traits, because if you were to rely on any one individual trait, you would find out, well, this group of worms is an exception, or this group here. And so in general, I'd like to move on, but deuterostomes do seem to be a group, but it's a kind of conglomeration of some embryological traits, some traits in adults, some genetic traits, which causes us to do this. And some lineages, may have lost something that their ancestors had. In any case, echinoderms are deuterostomes. Echinoderms were once enormously successful. So when the great radiation of large animal life begins in the Cambrian period, echinoderms are very richly there. Unfortunately, many of those groups have then become extinct. So echinoderms have an incredibly rich fossil record, especially early in the history of animals, with crinoids and eocrinoids and blastoids. And a lot of these groups are either extinct or largely extinct. So here's, these fossils. These are of what are called crinoids. They kind of look maybe like a starfish with feathery arms, but they're on a stalk, a stem that attached it to the bottom. So here's parts of the stalk. So they were stalked animals. They might have looked more like plants, if you had stumbled upon them, but they would have been like starfish-like animals on top of a stalk. They were once extremely common. Today, there are a number of groups of echinoderms which are still with us. The starfish, the sand dollars, the sea urchins, the sea cucumbers, and others. So here I have some images of a couple. So the starfish, which can use their arms to say, grab a mollusk and pull open the shells. They can then evert their stomach and digest the mollusk while it's in its shell and suck up the remains. Here are sea urchins whose spines are defensive mechanisms, sand dollars, and others. And once again, so I have a few videos on these. So echinoderms are a group of invertebrates which are more closely related to the chordates and the vertebrates than they are to, say, arthropods, etc. Now, they have completely reworked their body plan, because bilaterians in general have this obvious head and are bilaterally symmetrical. They have replaced this with a symmetry which is kind of more based on, say, a 5-point system when you look at starfish, although there are certainly other, some starfish have far more than 5 arms. And so sea urchins, sand dollars, etc. These are echinoderms. They are a group of deuterostomes. Now, they are so highly modified that they're not a good example of what the early deuterostomes might have looked like. And so I go into, there's the stomach of the starfish, there's the digestive gland. So they secrete digestive enzymes into, say, the clamshell that they're ingesting, and then absorb the nutrients through their mouths. Oh, and then just one other thing. Not only are echinoderms fairly common in the oceans, but so are their embryos. And so when we think of zooplankton, the small animals which might be nourishing small fish, this would include, crabs have very tiny, larvae, so they would be in the zooplankton, as would starfish embryos. So starfish sperm and ova can be released into the seawater. Fertilization occurs away from both of the adults, and then the embryos, start to develop. And so once again, this can be a part of the zooplankton, and an important food source for small fish. But once again, echinoderms are so highly modified that these hemichordate worms are a better example of what an early deuterostome would have looked like. So these are deuterostome worms alive today. Note once again that worms are a term we use to describe lots of unrelated things. So flatworms, roundworms, segmented worms, priopulid worms, and others are not at all related to these, which are deuterostomes, closer to vertebrates than they are to any of the others. One of the features is they have a throat with slits. This helps them feed, so they can suck water in continually, and the water goes out their the slits in their throat. But while they're doing this, then any food particles can get stuck in mucus, and cilia can sweep it towards the stomach. So they get to eat a meal, right? Because they create this suction, water coming in here, leaving there, and that allows them to feed. Now, this is a feature of all of the chordates, as we'll see. Even we vertebrates have this feature as embryos. Fish will use it in the gills, and I'll get to that in a second. And that ciliary response I still use. I'm still sucking in things, but if there's dirt and debris, I still catch it in mucus and I sweep it into my stomach with cilia. Now, I'm not using it to eat the way that these things did, but nevertheless, it is a common shared pattern. Later deuterostomes, which weren't hemichordates, but rather chordates like this one, put blood vessels in this area. And if you put blood vessels in here, then as that water keeps leaving, here are the blood vessels in a shark, the blood can absorb oxygen from the seawater. In other words, you're using it to get oxygen, you're using it to breathe. So now these aren't simply slits which are used for feeding, they're now gills which can be used for breathing. Now, this is a feature of the chordates, this throat with slits. Obviously, fish have gills, but this is a pig embryo. I notice that pig embryos develop this throat with slits. They change their throats because they don't have gills. Even in humans, we develop a throat with slits. Some individuals are born with a birth defect where this slit is open and has to be closed surgically. And so this was a feature of these early deuterostomes, which then became important for the chordates. And as I said, we still use mucus to catch debris, and we sweep it towards our stomach, but it's no longer a feeding mechanism, but rather a mechanism to protect our lungs. Okay. Now, from these early chordates, we would then get invertebrates which started to look more and more like fish. And then from things that look more and more like fish, we would get the first fish. Now, I know this sounds odd because when you think of invertebrates, especially say wormy ones, they don't look like the vertebrates, like ourselves. And when we think of fish, you're thinking of bass or trout or marlin or something. But the first fish looked like this. Right? The first fish were only an inch or two long. They had no jaws, they had no bone, they had no pectoral and pelvic fins. They were small, and actually, some of them, we could argue that they were still invertebrates. They didn't have elements of a vertebral column. And so from these chordates, all right, you start to have invertebrates which have some features of the fish, all right, and the vertebrates. So they start off as invertebrates which have some vertebrate features, and then the earliest vertebrates still look an awful lot like these early chordate fish. So just because this is a fascinating question, how could an invertebrate have some but not all of the features of the vertebrates? Well, I'd like to elaborate on that. But that's what we call, the chordates. So hemichordates, they have some of those features, but then chordates, which do not include the acorn worm, but include the lancelet, they have even more. So developing vertebrate features developed in stages, and so much so that this chordate here from the fossil record, the lancelet alive today, and ourselves, we are put together in one big group called the phylum Chordata. So just as there is a phylum Arthropoda, which unites the arthropods, a phylum Mollusca, which unites the mollusks, vertebrates and these things are classified in a group of phylum Chordata because they share features. Once again, they share a throat with slits, all right, which is not so important in us because as embryos we have to remodel this. But whether it be these early chordates or fish, this is important because this is where the gills form. There is then a notochord, a support rod, which goes down the back. This is, if you want to think of it this way, the precursor of the vertebral column. Before there was a backbone, there was a notochord. Now, these primitive chordates developed a notochord. The early fish still today, and the early fish, I mean, most primitive fish alive today, still use a notochord. And then even human embryos develop a notochord as embryos. In invertebrates, the anus will go to the tip of the body. The intestine runs the whole length of the body and ends here. Whereas in the chordates, the digestive system would end here and form and not reach the tip of the tail. So there would be a post-anal tail, which would only now have, say, muscle and skeletal elements, which can be used for propulsion. So that's not a feature of mollusks or arthropods or annelids, but it is a feature of the chordates that the intestine doesn't run to the end of the body. And so here we can see here's a pig embryo with slits in the throat. Here's a chicken embryo with slits in the throat. That even if the adult chordates, like we vertebrates, don't have some of these features as adults, we did as embryos. When we were developing as embryos, we had a much more prominent tail, and some humans can be born with elements of an external tail, which are then removed surgically. The notochord is the primary support structure of the primitive lancets and the primitive fish, but even we vertebrates have this support rod, a notochord, as embryos. And so there are these features. There are certainly others. If we were to start talking about, some of the immune proteins, the clotting proteins, we have a similar setup of our embryonic blood vessels, where our ventral heart sends branches of the aorta to aortic arches in the gill region, which then fuse to make a dorsal aorta. The point is, arthropods don't have these features. Mollusks don't have these features. But vertebrates, at least vertebrate embryos do, as do these chordates. So we're put together in this common phylum. Those slits with throat that have the branches of the aorta there to breathe, obviously very important, because here you can see these serve as the gills for fish. So the early chordates had the precursor of fish gills. They weren't fish yet, they weren't vertebrates yet, but nevertheless they were getting some of the vertebrate features. And so the features of vertebrates evolved in stages, right? And so there's a lot of these features which unite the chordates. So I'd like to just quickly overview a couple of other ones. In things like arthropods and mollusks and annelids, the nerve cord, the spinal cord, is solid and it goes on the ventral side. Whereas in the chordates, the nerve cord is hollow and it goes along the back. So here you can see a pig embryo is hollow, and in humans, we still have a hole in the middle of the spinal cord called the central canal, and the brain starts off as being hollow. And even as adults, we have ventricles in the brain filled with cerebrospinal fluid. So this is a feature of the chordates, which unites them and is different from what we would see in the protostomes. And so this is just a quick overview. There are many of these features, especially once we start getting into the genetic components, which show that in the great family tree of life, there were ancestral chordates from which modern chordates arose, and then also the primitive chordates, and then also the vertebrates. And as is pointed out in this, ciliary video, these offered the chordates an advantage because the notochord, allowed it to these early chordates to move in an S-shaped pattern, the way that fish do. And even if you look at salamanders or snakes on land, they're still moving here. The arthropods, the annelids, they don't move this way. So this notochord gives this support rod, which lets these primitive chordates move in a way similar to fish and many other vertebrates. The postanal tail is just for propulsion. The gills let the animals get more oxygen, and thus let them support faster swimming. And so these early chordates would have been better able to, in this example, evade a predator and swim faster because of getting more oxygen, of having that notochord support rod and having that post-anal tail. So this was an advantage for the early chordates, and then some of them would then evolve into early fish. So these early chordates had gills. Fish have gills, and then tadpoles can have internal gills, and there can even then be external gills in amphibian larvae as well. Here, if this helps you remember the things that chordates have in common, I have a chordate song, all right, which just kind of sums that up, because in addition, chordates have a homologue of the liver, including the hepatic portal system. There's a homologue of the thyroid gland. Right, and so, a ciliary song. So if that helps you remember the things that chordates share. And so this series of videos was meant to summarize the groups of invertebrates before we start looking at the diversity that there are in vertebrates. The fascinating thing about the deuterostomes is that they share features with the vertebrates. So look at this leech, all right? It's not moving in that S-shaped wriggle because it doesn't have a support rod going around its back. So the types of movement which you find in the non-chordates is different because when the chordates, once you get that support rod going down the back, that notochord, and in later vertebrates, this vertebral column would then form around it. This allows a new type of movement, that S-shaped pattern, which is, say, how the fish swim. But this feature of fish evolved in primitive invertebrate chordates, which had some but not all of the features of fish. So as we end, the survey of the invertebrates, we end with the group which is most closely related to the vertebrates, and the features of the early fish evolved in stages through these hemichordates and then chordates, which had some but not all of the features of the vertebrates.